There are some key features I would like to highlight here that make Scailyte's analytical approach unique. Considering the high complexity of multi-omic single-cell data, the automated pre-processing pipeline and the integrated AI algorithm of ScaiVision offer an unprecedented speed in extracting differences from datasets, delivering results within a few weeks, and combined with the richness and comprehensiveness of high-throughput single-cell data, it reaches an unmatched discovery success rate. Upon a benchmarking experiment, our data scientists demonstrated the robustness and reliability of its discoveries when compared to other single-cell analysis tools. One very important unique feature, thanks to the shallowness of the neural network, is its explainability. ScaiVision's discoveries do not restrict themselves to a trained AI model. Next to predicting the condition of a new samples or patients, we can go back to the model and extract a list of the cellular and molecular characteristics responsible for the prediction, and this list becomes the basis of a clinical assay prototype. This feature makes the algorithm a perfect fit for the discovery of clinically relevant biomarkers. Now, all of these features sound really nice, but if you are to believe me, I have to show you some numbers behind them. Let me focus on the discovery potential of ScaiVision. We use publicly available datasets to demonstrate the superiority of our approach versus the current standard of single-cell RNA sequencing data analysis, particularly for the discovery of clinically relevant biomarkers. What you see here is a single-cell RNA-seq data from 24 CAR T-cell infusion products used to treat lymphoma patients. A common severe side effect of this treatment is the development of neurotoxicities. By comparing clusters of patients that develop high versus low-grade neurotoxicities, the authors identified a rare cell population they call ICANN-associated cells, or IACs, which was enriched in high-grade toxicity patients. However, the authors state that they failed to find any significant differences between the CD4 and CD8 T-cell compartments of these patients. If you would like to use this cell population to predict the development of high-grade toxicities in the treated patients, you will have 58% sensitivity. We fed the single-cell RNA sequencing data together with the neurotoxicity endpoint information, and by using representation learning, ScaiVision learned from each cell separately which profile corresponds to patients that would develop high or low-grade toxicity. It identified the same population of IACs as described by the authors, but in addition, it included cells from the CD4 and CD8 compartment, which together comprised a more complex signature, which was able to stratify high versus low-grade toxicity patients with 100% accuracy. This example highlights the advantage of keeping the single-cell resolution throughout the analysis and the sensitivity of ScaiVision to find differences in two datasets, and particularly single cells and complex signatures. To translate this into a clinically relevant setting, if Scailyte's approach would have been applied in the original R&D setting, these patients would have been identified prior to treatment for better risk assessment. Scailyte's analysis would also have contributed to de-risking the clinical trial and allowed for a more successful outcome.